Let's give it another few seconds before we get going. I think, yeah, maybe let's get going while people are still joining. So hello, everyone. Welcome to this iOS seminar. My name's Chris Cooney. I'm one of the iOS committee members, along with some of the other people you see here. And just a bit of background in case this is your first seminar. The iOS network aims to promote the integration of speciation research and also the integration of the speciation research community. And one of the routes through which we are hoping to do this is via this regular seminar and discussion sessions and series, which are, of course, open to everyone from students to established researchers and non-scientists alike. And so hopefully you can see on the slide the dates and topics of our upcoming sessions, including today's session. So please do mark your calendars for these sessions. And more details in due course as we announce speakers for these sessions. So to that end, we would like at this point to also ask for your suggestions of speakers for our seminar series. We're always looking for ways to diversify the speakers that we invite and the topics that we address. And we'd really like to hear your suggestions for speakers and topics that you'd like to hear from. So we should be able to, and it's just there, we've created a Google form for you to be able to post your suggestions in there, and we'll record them. And obviously, if one of your speaker suggestions is taken up, we'd also warmly invite you to co-host the seminar with us if you like. So at this point, I'll just hand over to Roger, who's going to just briefly talk about another point of interest.
Yes, this is just to announce that the iOS network is supporting a meeting organised by the Linnean Society of London that will be held at the Linnean Society's rooms, a one-day meeting on the 11th of April 2024. The idea being to cover different views on the process of speciation, all the way from a philosophical viewpoint through paleontological viewpoints to genetical and microbiological and so on. In-person places are limited, but hybrid online attendance will also be possible. So the link for registration's on the chat, and it will be also in our website and coming out on Evil Deer hopefully tomorrow. So we look forward to seeing some of you there in person or virtually.
Fantastic. Thanks, Roger. So without further ado, let's get on to today's seminars. So these set of speakers are addressing the topic paleontological views of micro and macroevolution. And it's my pleasure to be able to introduce Dr. Josef Uyeda. Josef's an assistant professor in biological sciences at Virginia Tech. And Josef's background is in herpetology, quantitative genetic modeling, systematics, and phylogenetic comparative methods. And work in Josef's lab focuses on establishing better connections between microevolutionary processes and the models that we actually use to understand trait evolution at macroevolutionary scales. And Josef and his team have produced some really landmark pieces of work in this field, in the field of macroevolution and phylogenetic comparative methods. And we're very grateful to Josef for being with us today, and we're excited to hear about what you have to say. Thank you very much.
Thank you, Chris. So it says I can't shift it. There we go. Okay, does that look okay? Everyone can hear me? Great. All right, so thank you for the invitation to talk here today. I am not a paleontologist, and I don't think Michael is either, but either of us would claim to be. But I want to talk today about how we think about macroevolution, and in particular, the evidence we use for it. So that comes from the fossil record, that comes from comparative data and phylogenetic data. And many other potential sources of data. So how do we understand macroevolution? And are we using all the evidence? And so what I like to start with is going to my slides work here. The original Felsenstein 85 paper that introduced independent contrast, because I think this intuitively demonstrates what we think of as macroevolutionary evidence for a relationship, for example, here between X and Y. We can see that this, what initially may look like good evidence, is actually not good evidence for a relationship between X and Y. And it's very intuitive, and we understand just based on this simple figure how phylogenetic data can influence our understanding of the evidence in these, in this example, trait values for X and Y. Since that paper, of course, there's been an explosion of methods of increasing complexity and sophistication for testing all sorts of comparative hypotheses about diversification of both traits and of lineages. But if you're familiar with this field, you probably also know that there's been lots of papers that have started to question whether we can actually make the sorts of conclusions and inferences that we're often after as evolutionary biologists. And the fundamental problem is that we can't necessarily do experiments at these million-year timescales. We're limited to these observational data sets, and we're often limited in the number of species. So how can we make sense of these patterns with our limited observational data? And that's kind of the challenge I see facing phylogenetic comparative methods and studies of macroevolution, and what I want to talk about today. And I'm going to talk about two projects that I've been involved with. And the two themes for how we can deal with this sort of complexity is first to think about not just more species and more comparative data, although that's always good, but to think about different types of data and establish new types of links between our models and those data to better understand the process. And then in the second part, I'm going to talk about how we can confront our evidence with our causal hypotheses. This is something we don't like to do because it's observational data and we're afraid of the word cause, but I'm going to show the benefit of confronting that evidence with our actual causal hypotheses. Okay, so to jump right in, I'm going to start with this first one here. This is a project that I've done in collaboration with Dr. Carrie Mongol, who's an expert on primate evolution. She's an anthropologist at Stony Brook University, and knows all about the molars and the dentition of primates and how they evolve. We brought on Fabio Machado, who was a postdoc in my lab, is now assistant professor at Oklahoma State University, and he did a huge amount of work on this project. And so why primate molars? Well, primate molars are cool because they have a lot of data. We are interested in primates as humans. We are primates, and so there's been a ton of work. So we not only have comparative data, so many species for which these traits have been measured, but we also have good phylogenetic trees. But in addition to that, we have a very robust fossil record. Things like primate molars have a very good fossil record. Many the fossil record. We even have modern time series on the generation time scale where people have studied molar evolution. And we have groups that have worked on the quantitative genetics and evolutionary development of primate molars as well. And all of these pieces have the potential to inform our macroevolutionary modeling and link to different aspects of the process, different windows into the process across time. All right, so what is the developmental program for molars? Well, they develop sequentially. So there are 3 molars in most primate species. The first molar develops first, the second molar develops after, and the third molar develops after that. And this process is regulated by an inhibitory cascade. And what does that mean? Well, that means that the first molar develops first, and as it's developing, it actively inhibits the second molar, which then buds off of it and develops into the second molar. And so the larger the first molar, the more inhibition there is, and the resulting smaller size of the second molar. And so as this process progresses, you get this very the influence of the size of the first molar on the second and the second on the third. And then that also results in an increase in variation from these, because the number of genetic factors in the first molar influence the second molar, and then you get more variation building up in this sequential sequence as you go down the molar line. And so the inhibitory cascade model predicts this sort of relationship between molar sizes, particularly their ratios. So if the first molar is larger, then the second and third molar will be smaller. If the first molar is smaller, then the second and third molars will be larger in this kind of very predictable way. And so we sought to look if the relationship to the macroevolutionary rates was captured by this kind of micro to macro prediction, that the amounts of microevolutionary variation available might predict this macroevolutionary rates and patterns. And so we made this prediction that as you go down the molar line, you would see increasing rates of evolution. And that's exactly what we saw: increasing amounts of variation, increasing rates of evolution in this paper by Carey in 2022. But we wanted to do more and really see if knowing about this link between this microevolutionary variation and macroevolutionary rates could help us better understand the history of selective factors that have influenced the diversification of this group. And we were lucky enough to link up with Graham Slater and Anna Wisniewski at University of Chicago, who had just put together this enormous phylogenetic tree of primates that included not only nearly every extant species, but nearly every fossil species that we know about. And we were able to target these fossil species in the literature, and by collecting museums, looking at museum specimens or photographs, to collect data for 558 species. Over half of them are actually fossil species. So this is a really densely sampled fossil data set. And what's cool about this, and what makes this really exciting to me for our ability to fit these macroevolutionary models, is I think it should be one of the more powerful data sets we have. We're not doing something like trying to guesstimate the body size of extinct taxa from their molar. We're actually studying one trait, the same trait, set of traits, in a multivariate context across all of these lineages all together at once, and we have all these different sorts of data coming together. And so what Fabio did is he linked these kind of state-of-the-art macroevolutionary models for these sorts of continuous traits. So there's a package called PCM-FIT that was introduced by Venneli Mitov and colleagues. This is kind of the Death Star of phylogenetic comparative methods for Gaussian models, because it can do everything. It can do OU models, it can do Brownian motion models, and it can do them in a multivariate context. And it can also find shifts and heterogeneity across the tree and split the model and do all sorts of really cool things. But if you've tried to use it, it can sometimes do too much, and it can be very hard to constrain or interpret the models that come out of it. And in some cases, they may be biologically unrealistic. These models are very complex. They need constraints. And so what we sought to do was to simplify these models by linking them to these outside sources of data. So we looked to the work of Leslie Lusko and colleagues, looking at the quantitative genetics of molar evolution in primates, especially in baboons. And we used those data to link to the parameters so that we could constrain these models and test models where the macroevolutionary parameters are linked to the microevolutionary genetic variances and covariances. So how do we insert the Evo-Devo into this? So I remember when I was a postdoc talking to Matt Pinnell on like, Evo-Devo is supposed to be the solution to macroevolution. And he was like, isn't it funny we never use it in our macroevolutionary models, paraphrasing. And I think, there are certainly some people that do, but by and large, we don't use a lot of Evo-Devo in PCMs. And so where does it show up? So one of the places that it showed up for us here is, even for this relatively simple set of traits, there's multiple options for how we measure the data. We could make 6 traits, the length and width of each molar, and have the covariances between them, or we could define the areas. Or if we know about Evo-Devo, then we could use the inhibitory cascade characterization of the important space for these traits, which is the ratio of the first molar to the second and third molar, which is informed by this developmental process. So I call these first two the naive measurements, because these are the ones that we probably if I just had some calipers and some teeth, this is probably what I would do. But if you knew about the actual process of how these molars were produced, you might use this second one. How does that influence the models that we fit and what we learned from our data? And so here's the phylogeny, and here is PCM-FIT finding these different regimes, these differences between the fossil, strepsirrhines, haplorhines, and this is the results of this paper that's out now in Nature, Ecology, and Evolution. And I won't go through all these different models, but what I want to highlight is these first two categories are our naive measurements. And the best fitting model in these cases are these relatively complex models. I want to point you to the number of parameters, 74, 26 parameters. These are very parameter-rich models. But in our inhibitory cascade model, the best fitting model was a relatively simple model where it only has 7 parameters. And how did we accomplish that? It's by constraining the macroevolutionary parameters to match the microevolutionary parameters, to be proportional to those microevolutionary parameters. And the best model in this subspace identified by the inhibitory cascade is consistent with genetic drift around a stationary optimum, a very simple OU model for the evolution of these traits, where the genetic variation and the expected change from genetic drift over time is well aligned with this underlying adaptive landscape. And so by understanding the EVO-DEVO, we were able to find this much more interpretable and subspace of the morphospace that aligns to many of the predictions of microevolution. So I don't have time to go into all the details of this study and interesting questions we looked at, but EVO-DEVO guided measurements seem to have identified a simpler evolutionary model and made it easier for us to connect to meaningful measures of process and evolution at this scale. Okay, so I haven't talked about speciation yet, so I want to make sure I talk about that to this group. So how do we confront our evidence with causal inference? So this is another project that's been led by primarily by 3 postdocs, Laura Alencar, Orlando Schwery in my lab, Saul Domingos Guerrero, and Martha Munoz at Yale is not a postdoc in her lab at Yale. And the question that we're seeking to answer is, what are the causes of diversifications in pleurodonta lizards? Okay, so this is the kind of typical adaptive radiation sort of question. And how do we usually tackle those in macroevolution? We can tackle them with things like SSE models. So where we think that diversification might be related to traits. And so we might apply a BISI or a MUSI model. Here I have a BISI model where I have a binary trait that I think affects diversification or speciation rate. And I have 2 different parameters, depending on if you're state 0 or state 1. And those might, in the phylogeny, play out such that, state 1 looks like it has a higher diversification rate in this case here. But we also know that there's more complexity in macroevolution. It's millions of years it's basically all of evolution, right? Millions and millions of years. Everything happens over millions of years. So there's other factors besides our focal traits and factors that are probably playing a role, right? And sometimes our focal traits may not actually trigger diversification where they do in other cases because of some contingent factors, right? And in order to study these, we've learned that it's important to consider the possibility of hidden states. So we have things like HISI, which allow for hidden states or latent traits in the phylogeny, such that in this case, for example, we've expanded to 2 hidden states per observable state. We now have a 4-state model, and maybe there's a hidden state within our trait 1 where there are actually 2 different groups. One is diversifying faster than the other because of some unseen factor or whatever. Okay, so this is kind of the model set we tackled this question with in the pleurodonta. And so we have a radiation of over 1,000 species, of which we have data for about 733. And there's a handful of factors that we hypothesize may be responsible for increasing diversification or different radiations in this group, ranging from parity mode, are they viviparous or oviparous? Do they exist on mountains, or particularly the Andes Mountains? There's North American, South American radiation, a lot of species in the Andes. Arboreality, diet, islands, body size, a fair number of candidates, right? But in the grand scheme of things, not that many possible factors that we're really examining here. I'm going to zoom in on two of these, parity mode, because it's thought to be an important potential key innovation for diversification in some habitats. And that's because of its dependence on the environment. The idea that viviparous species, by retaining their offspring, can thermoregulate better and potentially colonize areas where there's not enough, environmental temperature to maintain incubation of those eggs and development of those eggs. But it also comes at a cost, right? Because you're carrying around these offspring for a long period of time as a gravid female lizard in a particular environment. And in particular, these traits are thought to be tied to these sort of mountain environments, right? Because maybe being viviparous is beneficial in mountains where it's not as warm relative to the tropical lowlands, and you can't just leave your eggs somewhere and expect they'll get enough warmth to develop properly, right? Okay, so what we're really envisioning here is adaptive radiation through ecological opportunity. So maybe some mountains originate, and you either have viviparous species colonizing those and then diversifying, or you have oviparous species colonizing them, transitioning to viviparity, and then speciating and having a high diversification rate, right? Okay. So I've kind of jumped ahead to the MUSI model, which is the multi-state, state-dependent speciation and extinction model. And here we have two potential factors that we think are affecting diversification: the Andes Mountains and parity mode. And those are affecting, and putting speciation rate, you can put speciation rate or diversification rate. And we make combinations of species that are either not in the Andes and oviparous, or in the Andes and viviparous, right? And we estimate these different parameters and we see the model fit. So Orlando did this and got these results for the diversification rates. And you can see that the viviparous species on the Andes have the highest diversification rate, intermediate values for these guys, and then oviparous non-mountain species have the lowest diversification rate. And so, okay, that seems like a sensible result. But we know that there's the possibility that there are other latent factors that are potentially confounding our results and giving us incorrect inferences in this regard. And so if we add hidden states to parity mode and just look at that in isolation, we do indeed see that there are two hidden states for viviparity and there are two for oviparity, and one of the hidden states for oviparity is actually higher than the diversification rate for viviparity. And if we combine them all into a mu-HISI model, it gets complicated and it's a little bit messier, harder to interpret it. We have less data per parameter. It's a more parameter-rich model, but overall the trend seems to still maintain that viviparous species tend to have higher diversification rate. That seems to be the best predictor. The Andes Mountains also probably play a role, and there's probably some other hidden factors that we haven't quite gotten yet, but we can deal with those, right? And so this is our conclusion from this traditional approach. There's latent traits. Viviparity is number one. Andes also plays a role, and it seems like there may be synergistically acting to promote diversification. Great. So we've got our paper, but we started looking at the reconstructions and really looking at our data. And one of the first things you'll notice is that the hidden states don't appear randomly across the tree. Instead, they're kind of localized in this one clade, the Liolamidae. And so this is a radiation that is primarily in the Andes, but South America, but they've secondarily come out of the Andes as well. And you can see that the hidden state for oviparous and viviparous species is localizing in this clade. And this clade actually provides the perfect test case for our prediction of our primary conclusion, right? That the Andean viviparous lineages diversify faster than Andean oviparous lineages. And this is testable because there's been multiple transitions from oviparous to viviparous lineages within this clade. And so we can do just like a simple sister groups comparison. For example, comparing these red viviparous lineages to their sister oviparous lineages, and ask the question, are they diversifying faster? They don't really look like it from my eyeball test, my eyeball estimator. And if we actually look at this, it seems relatively unlikely that the viviparity is actually causing high diversification here because the oviparous species are also virtually the same diversification rate. So what's going on here? Is there a potential that we actually have evidence in our data that is refuting our primary causal hypothesis for what we think is driving diversification? Okay. And so what is causal inference and how do we test it? So I'm going to give a brief tutorial on how causal inference is done, one way that it's done, and that's through things like path analysis, where we use graphical models, where we have a hypothesis that maybe A causes B and then B causes C. Importantly, we don't have an arrow here going from A to C. And so under this causal model, you might predict that you would see this sort of relationship between A and C. But if you measure B and account for B, you don't expect A to account for anything else in the data, right? Because the entire effect of A is mediated through B, and if you have B, then having A doesn't actually help you at all, okay? So we actually would expect that conditioned on B, there will be no relationship between A and the residuals of C on B, right? If we instead found this would suggest that A actually does still have an effect. If we had a significant effect, we could reject our previous causal model, and we could potentially, so this causal model here, we can reject that if this is a significant relationship because there should be none under that particular causal hypothesis. However, these two causal hypotheses as alternatives are still plausible, right? They still could be explanations of this process because both of these have a direct effect of A on C as well as a potentially an effect of B, possibly mediated as a mediator from A. So we can do the same thing with the Felsenstein scenario. And this actually allows us to do something that's relatively hard in causal inference, which is to deal with latent traits. And so typically it's really hard to deal with hidden or latent traits. But if we think about this as something happens in this difference between these two clades, we can think about phylogenetic confounding. And that's what I think is really intuitive about the Felsenstein example. There's some latent trait that maybe affects both trait 1 and trait 2, and it happened in this split between these two major clades. And that explains why there's a significant regression between these points. But within these groups, there's nothing. And so there's actually not a strong relationship between T1 and T2 in this case, right? And so can we apply this causal logic to pleurodonta diversification? So this DCEP test is all about predicting the conditional independencies of traits for a particular causal graph. And so let's see. So here we have, so I'm not going to go into too much depth on how you do this, but the way you read this is that we have some latent factor that we think is affecting the liolamus that's causing them to diversify at a higher rate. And if this is the causal model, then we predict that L is conditionally independent from V, which in our case is parity mode here. Or we could have this causal model and we would predict these conditional independencies. Or we could have this causal model and we would predict these conditional independencies. So I'll describe these in the next slide where we actually test that. Okay, so how do we go about doing this? Well, the first thing we did was to identify the major shifts in our data using what I call phylogenetic natural history tools like MISSI or BAM that find shifts in diversification in the tree. Where are the major shifts? We then combine those data-driven identified shifts, these kind of big factors that influence diversification, with our MUSI states to create an 8-state MUSI model. Okay. And what are the results? We estimate net diversification from those, and then we compare them to the predictions of these, the tests based on these different causal graphs. So in this one, we predict that after conditioning on viviparity, diversification and this liolamus clade shift should be uncorrelated to one another. And so we have these 4 major shifts that we've identified. Here is the major background shift in yellow, and then I'm going to focus primarily on the liolamus because I don't have time to get to these other shifts here. But we can see that after conditioning on viviparity, that both oviparous here and viviparous species have elevated diversification rates relative to the background. And so we reject this prediction that they should be uncorrelated under this causal graph. However, it is consistent with this model where there's a factor that increases diversification in the Liolaemus, and if viviparity has an effect on diversification, it's much weaker than what we initially saw, right? There's only this slight difference between the Liolaemus oviparous and viviparous species, just like I showed you before. Contrast that with the Andes Mountains. If we replace viviparity with the Andes Mountains, we predict that after conditioning on the clade, that this diversification of oviparous or of non-Andean lineages should be the same as the background, and indeed they are. Okay. So we can't reject that causal hypothesis, but we can reject the causal hypothesis that Andes doesn't have a direct effect on diversification, and that this latent factor is causing both, because there still is a relationship within that group of the effect of being in the Andes Mountains or not. And I know Michael has a great paper on bioRxiv that looks at this in the Liolaemus a little more fine scale. Okay, so we can't reject this causal model. And this leads us to a kind of reversal of what we initially had concluded using our traditional approach, where the Andes and viviparity influence diversification synergistically. It appears that viviparity does affect diversification, but it's a much weaker effect than we actually estimated. And the primary effect is this Andes Mountains, which, you know, if you look at diversification globally, Andes probably loads pretty high on PC1 of that variation. And so the Andean Mountains appears important for Liolaemus as well as this Fimaturus. And why didn't we get this with our original Musi model? I used a Musi model to fit this. Why couldn't I have found that to begin with? Well, it's because there's other stuff going on. And so the problem is that if we think about our model set going from there's only one thing and we've measured it, to have maybe some hidden states, a couple things going on that we haven't measured, to a handful of things going on we haven't measured, this model gets very complex very quickly. People are testing, you know, tens, if not, if you go this direction, potentially hundreds of models you could end up having to test. And it's very hard to deal with it. Maybe once we get down to things that happen all the time, we can just do something like a PGLS and it might be a little easier. But in this intermediate zone, this is where we often are. There's a handful of things that make it complex. It's very hard to find the exact adequate model. And causal inference, I think, is a way to get there more directly by really looking at the evidence in our data and the predictions of our causal hypotheses. And so that's my pitch for how we need to address that. So the take-home message, our models are complex, but getting different types of data may help reduce complexity and elucidate process. And instead of expanding our ever-growing model set, just kind of everybody apply 300 models to your data, can we use the logic of causal inferences and the knowledge we have of our causal hypotheses to more directly evaluate these macroevolutionary data sets to test and understand what are the likely or plausible processes underlying that diversification. So with that, I'd like to thank the organizers. I got on the trend of getting the Google Scholar word cloud, very unsurprising for me. And thank you.
Thank you very much, Josef. Brilliant talk, very interesting. Do we have any questions? As Carol mentioned, if you do have questions, please type them into the chat and we'll put them to our speakers. While we're waiting for that, I had one actually, Josef, if I may. I was interested by, maybe this is a very simple thing, but in the primate molar study, you contrasted the direction and the kind of shape of the genetic variance ellipse with the adaptive landscape ellipse. So could you just say a bit about how, particularly the adaptive landscape was estimated in that example?
Yeah, so we estimated that from the OU model, right? So the OU model predicts you have this kind of Gaussian attractor to this adaptive peak in the multivariate context, and then there's drift around that adaptive peak. Okay, so those lines that we drew there were from our estimate of the kind of uncertainty in that adaptive landscape taken directly by translating the parameters of the kind of selection component of the OU model, if you want to interpret it that way, or the macroevolutionary landscape component of the
Great. Just maybe a quick follow-up while we wait for other questions. Is it feasible to independently estimate the adaptive landscape and then see how well either the kind of the trait variance, the trait diversity fits that independently measured adaptive landscape, or even the kind of the model fitted parameters recapitulate that? So I have actually no suggestions about how you would do that, but in the primate molar example, could, in theory, the adaptive landscape be estimated separately and then used as a kind of prior or a sanity check on the kind of adaptive landscape that emerges from the model fitting, if you see what I mean?
Yeah, absolutely. So, I mean, there are microevolutionary estimates of the adaptive landscape that have kind of, come from the work of Russ Landy, quantitative genetics, estimating selection surfaces, and then translating that to an adaptive landscape at the population level. Of course, you can only estimate that adaptive landscape for the window over which the variation in the population exists, right?
Mm-hmm.
And I was actually really surprised by our results with the primate molars, because I am usually careful when I apply an OU model, even though a population adaptive landscape with genetic drift will result in an OU model, I usually don't think of it actually measuring that type of selection, but instead of some macroevolutionary landscape on which adaptive landscapes themselves move. And so I was actually really surprised to see that the parameters matched really well with what you'd expect from just a direct translation of a microevolutionary adaptive landscape to a macroevolutionary scale with the quantitative genetics parameters. I think the reason that happened and wasn't more complex is because we identified this subspace by this highly conserved developmental program. Those highly conserved developmental programs may be predisposed to exist for traits that have a very stable evolutionary history at the macroevolutionary scale, right? And so I think it helped us to identify a subspace for which that was true. And so I'm kind of excited to see, as we get more, can we integrate more of this developmental data to understand these different spaces and identify ones that have this sort of selection dynamics versus this sort of selection dynamics, and how that interplays with the evolution of development itself.
Very interesting. Thanks very much. So I see that we've had a question, but I wonder whether in the interest of time we should go to Michael's talk, and we can put that's a very general question at the end in the discussion, if that's okay. So I'll hand over to Sonal.
All right. Thank you all. So now for our second talk, it's my pleasure to introduce Michael Landis, who's currently an assistant professor at Washington University in St. Louis. I first met Michael as grad students. We were both grad students at UC Berkeley. And what I respected and enjoyed most about talking with Michael at that time is how creatively he applies new methods to really interesting empirical data sets. And some examples of this are the application of stochastic Levy processes to understand vertebrate body size evolution, developing Bayesian methods to understand the biogeographic history of iconic radiations like the silver swords, and newly funded work that's using deep learning methods to understand how viruses spread. So I thank Michael for joining us, and I really look forward to his talk.
Well, I've had people say they enjoyed talking to me before, but never respected those conversations. That's nice to hear. I also, Josef pointed out too many slides, but because we're bridging micro and macro evolution, I'm either off by a factor of 2, thinking on deep time scales. I'm not sure. But let's get started here. So the general theme of my talk is focusing more on biogeographic aspects to understanding how micro and macro perspectives are bridged using phylogeny and paleontology. And I'm going to have to take you the long way around the barn here, because I really want to start with biogeography and what that has to do with speciation and species richness. And later, I'm going to connect that to notions of fossil data, paleontology, and so forth. So just bear with me for a moment as I get started. One thing I think basically every evolutionary biologist would agree with is that evolution always has a spatial context, particularly when we're thinking at the level of species. For this talk, I'm going to focus mostly on biomes because I think biomes capture not just a space, but also a set of environmental conditions. And those conditions shape when and where species will evolve, disperse, go extinct, and diversify. For example, a species...
Michael, sorry to interrupt.
Am I muted?
No, you're not. We can't see your slides. I'm not.
No,
It's
okay.
a bit uncertain whether you were, just introducing the talk, but yeah, we can't see anything at the moment.
Nothing is changing for you?
No. Oh, there we go. Yes.
I can try it this way.
No, we can see your desktop now. So if you go
Yeah.
to presenter, then
But this did not show, it sounds like.
this is the first time we've seen this. So yeah, sorry about that.
Interesting. Okay. So
Great.
here's the long way around the barn and all that. Here's a cute picture of a polar bear. And basically, I just want to emphasize, if it's not obvious, that polar bears can't necessarily survive in any habitat type as such. If they're entering into a new habitat type, they might need to evolve new adaptations to survive there, or perhaps they're pre-adapted for those habitat types. And other types of transitions are very unlikely. They might not be capable of intrinsically adapting to that habitat type. Now, I also want to emphasize that we collect our fossils in space. And so these fossils might tell us something important about the historical biogeography of a group. And that itself can tell us something about why do we find more species in one place than another? What are underlying environmental factors that drive speciation and so forth? Much like what Josef was speaking about with the Andes and the Leolamus lizards. Because this talk is intended to bridge micro and macro perspectives, let's start with microevolution. I would say most microevolutionary models tend to focus on the relationship between genetic mechanisms for creating reproductive isolation between subpopulations, ultimately resulting in species. And as such, those models often really strongly rely on having genetic data. And for most species that are now extinct, we simply cannot get genetic data from those extinct taxa. When you're completely mineralized, there's almost no hope of getting DNA. So that's going to pose a problem when thinking about bridging these perspectives with paleontology. On the flip side, with macroevolutionary models, these often focus more on the broader geographical and ecological factors that might drive patterns of species richness. These methods also might include genetic data, but I would say they place a stronger emphasis on species richness data sets or range data. And some examples here might be equilibrium theory for island biogeography, unified neutral theory, progression rule, things of that kind. The framework I'll be using, much like Josef, is a phylogenetic framework to try to bridge this gap. This is a nice, I think one of the major first perspectives really bridging the micro to macro perspective is this work on phylogeography by John Avis. And the idea here is that we should be able to say something coherent about not only how genes evolve over time in space, but also how the species do. And I'll be focusing, so here's a little diagram showing how different disciplines in evolutionary biology map onto one another. And perhaps not surprisingly, phylogeography is right there in the center in his view of the world. I'll be focusing here mostly on the bottom part with macroevolutionary disciplines, but we'll be connecting to microevolution through these molecular genetic models of nucleotide substitution. Now, as I said, I'm going to focus on biomes to reflect biogeographic distributions and understanding the history of ancestral affinities for different biomes should tell us something about the conditions that might generate species, or at least allow species to persist for long periods of time. So if I gave you two separate phylogenies and I allowed those, and these are the exact same branch lengths, exact same topologies, exact same tip states. And depending on what history I think is more likely, it might give me a very different impression of where did different species originate. On the left-hand side, I have 4 new species emerging in a warm biome, whereas on the right-hand side, I have an even split between warm and cold biomes. So which of these histories is more likely? It really does depend on a case-by-case basis. So we don't have good general rules for explaining these macroevolutionary patterns. For people who don't really work on species-level biogeographic, clade-level biogeographic inference problems, I just want to give a quick tour of how we often think about this. One thing to note is that biome affinities are very strongly linked to geography in most systems. Here's an example with the Hawaiian Silver Sword Alliance, which contains the very charismatic silver swords down there in the bottom. The Hawaiian island, the big island in the bottom right in yellow, is the youngest but highest island, and it has the highest proportion of dry habitat types of any of the 4 islands in this complex. And perhaps not surprisingly, we also see the highest proportion of species in these high alpine deserts, which aren't really found in most other parts of the system. When we look at different clade, when we look at another clade, here's a group of legumes, Cisalpinia, we find that different biomes are associated with different growth forms. So for example, we find that basically all of the species that occupy savanna biomes form these woody vines or lianas. So there's some type of adaptive relationship between trait and ability to survive in a habitat type. I'll also mention that every clade is a little bit different. I just showed you 2 examples, which are fairly distantly related. Here are 2 sister clades, the mango family and the frankincense family. In this study, we basically find that the mangoes seem to be very capable of not only adapting to temperate, but also tropical, dry and tropical wet climates, but on top of that, being fairly widespread across a variety of habitat types, more capable of generalization. When compared to the frankincense family, which seems to display a lot more of what you might call biome conservatism, phylogenetic biome conservatism. They do not rapidly shift among different biome types. Now, I'm bringing this all up mostly to emphasize that the historical context matters a great deal when you're trying to understand the conditions in which different species originated. This review paper by Michael Donoghue and Erica Edwards gave 3 examples for underlying historical conditions that might have an impact on what biomes we find different species in. In the first case, we might have an enabling trait, such as a species gains some ecophysiological capacity to survive freezing winters. That trait, if that trait might allow them to basically survive in an environment that the other species can't. In the second case, we might have biotic interactions, such as a new set of pollinators enters a biome, such as a cold biome in blue. And that allows a species to expand its habitat range into this new biome. In the last case, you might have some type of geographical change. Either the species itself disperses into a new region, the biome where a species resides changes underneath it, and so forth, which can allow a species to transition into new habitat types. So there's lots of factors that might influence why we see different species in different biomes or habitat types. And here's where I'm going to return to this issue of paleontology and how to integrate it. With many macroevolutionary studies, out of matters of practicality, convenience, and just for the availability of data, we often are forced or inclined to estimate phylogenies and do reconstructions only using extant taxa. Josef gave some really compelling examples for how including fossil molars, for example, might give you a better perspective on things. I think with the Leolamus lizards, it would be great if there are more fossils for that clade, but they're very small, fragile species that just don't preserve well in the rock record, unfortunately for us. And what can happen is when, and I'm not at all implicating anything about any particular study, but one thing that can happen is if you don't have a lot of information about the past and you base your inferences on the present, you can end up with, on one hand, uncertain estimates, which are bad. You might have low accuracy for saying exactly what happened. What's even worse is you might have, with a biased result, something that contradicts what could have happened because we've overfit our model to only data that we can collect or have included in the study. And what I'll basically be focusing on for the centerpiece of this talk is how can we incorporate other forms of paleo data, particularly from the fossil record, to better constrain how these models operate. So you'll see a lot of similarities in my thinking and Josef's thinking. And you'll also notice that these types of decisions really come from some type of intimate knowledge with the clade you're studying, the biology of the system, and so forth. It doesn't seem to be as easy to come up with general rules for how to approach this problem, which I think as scientists either is frustrating or exciting, depends on your perspective. So for this project, I'll focus on a group of flowering plants by Burnham. You know, my background's in computer science, but during my postdoc, I was lucky to team up with this group of excellent botanists, and it really changed the way I think about how to conduct problems in phylogenetic inference. So let me basically parrot everything I learned from those 3 or 4 years. Viburnum is a group of about 165 living species. You can see that they have these nice showy flowers. They're typically insect pollinated. They also have these very colorful berries in frutescuses, which are probably bird dispersed, but it's not entirely known. The viburnum plants, they occur in a wide variety of habitat types, typically these mesic forests. And broadly speaking, you can classify them into 4 major groups: tropical, warm temperate, cloud forest, and cold temperate forests. And you can get a sense just by looking at these images what those different habitat types might entail for survival conditions. The way that we'll be defining these biomes for our purposes operationally is that tropical forests have low amounts of seasonality and essentially no freezing, even during the coldest months. Warm temperate forests have some seasonality and rare bouts of freezing. Cloud forests also have some seasonality and rare freezing like the warm temperate forests, but they have much more cloud coverage. And then the cold temperate forests tend to have high degrees of seasonality. They're more continental climates and prolonged freezing during the winter months. When you look at the types of leaves you find in these different habitat types, they also display some of the leaf traits that are very typical for those groups, such as, entire margined glossy leaves with drip tips for tropical in the tropical biomes, evergreen leaves at that. Whereas in the cold temperate leaves, you tend to get more deciduous leaves with deep dentitions and even some pubescence and so forth. If we then turn to look at where we find different viburnum species, different living viburnum species, that is, and we group them into different major geographical regions, but also the biomes they occupy, you'll see that there's a non-random distribution of both the species richness in different regions, which are the more artistic blobs in the background, and the proportions of species, or the actual counts of species in those different regions. So in East Asia, we have that's the center of diversity for living viburnum species. And we have a split of about 44 against 44 species in the cold temperate biomes and the warm temperate biomes. Southeast Asia is where live, with the exception of this one here in East Asia. In Central and South America, so there's some relationship between where these species live and what habitats they occupy. It's not entirely clear what that relationship should be a priori. Now, people have studied viburnum from different perspectives. This nice paper in New Phytologist by Beth Spriggs and others basically classified viburnums as being either in tropical or temperate conditions. And they reconstructed a tropical ancestry for the group. And their perspective was that perhaps, some of the deepest lineages in this phylogeny, like Clemenceae here, are have very strong tropical affinities. And the thought here is perhaps these are the last embers, these dying embers that survived a lot of recent extinction when climate changed after the Oligocene. Oh, here we go. Tropical diversification slowed down after the Oligocene. A contrasting view is one put forth by Frederick Lenz and others, where they incorporated other information from closely related species, Doxa and Cenodoxa, and they recovered instead a slight preference for a freezing origin for viburnum. They also brought in some interesting information from wood anatomy, thinking that these scleriform perforation plates, which basically can divide and break up air bubbles in drought in regions where you have a lot of low water availability, such as arid regions or regions that experience a lot of freezing. These perforation plates can basically prevent vessels from getting blocked with these bubbles. And one question is, now you have 2 phylogenetic reconstructions, very similar tip data, very similar phylogenies, and they come to basically opposite conclusions for what the origin for the clade is. So we came up with 2 different strategies for incorporating paleo data into these reconstructions to hopefully get at the bottom of the problem. Because if nothing else, it's a little bit unnerving if 2 very talented and smart groups of people who care a lot about the history of a group come to almost opposite conclusions. Now, I'm going to start with the first strategy, I guess that's why they're numbered, where we'll look at how can we include individual viburnum taxa to better understand the origin of the group. Where did these species first begin to originate? And how did species richness change over time? So with viburnums, we have basically 5 nice fossil pollen representatives coming from paleo assemblages. And here's what one fossil pollen might look like. An important feature of these fossil pollens is the exine, which you can see here, which is basically the matrix in which the seed code is encoded. Now, this fossil that I'm pointing at, 1A, occurs in the early Eocene about 48 million years ago at a paleo latitude of about 60 degrees north. This fossil is just one of a lot of fossils that come from the same assemblage, from the same paleofloral community. And what we did is we basically went through a series of papers that describe the location, the Princeton chert for that particular fossil and the other fossils that we could obtain, and tried to get a sense of what was the actual habitat type for that corresponding to that fossil when it was collected. So for 48 million years ago in the Princeton chert in the Eocene, we referred to these two papers et al. And Greenwood et al. And they used a variety of methods and included some images of how they think the paleo community looked. This represents a distribution of different genera and other major clades that were recovered from the assemblage. And you'll notice that some of these things, like Sabal palmetto, are to the right of this number 0, which is the coldest mean temperature in centigrade, so above freezing temperatures. Some of these lineages that are there are well to the right of this 0 degrees centigrade. Now, collecting this data set requires a lot of detailed paleobotanical work. That was not done by us. We relied on other paleontological researchers for that. I also want to emphasize that these types of data sets are very sparsely distributed in space and time. So not every fossil is guaranteed to produce similar information. But by basically looking at this analysis here on the right is called a nearest living relative analysis. And basically, because they look at the range of temperatures that living species can occupy and survive in, and they use that to basically predict what type of climate an ancestral species or an entire floral community could possibly tolerate. So if no living species within this, for this particular group are found in below 0 degrees centigrade temperatures, one conclusion that you could draw is that it's consistent with the idea that these floral elements could not have survived in that freezing temperature because no living relatives can do so. So these analyses bring in strong assumptions of evolutionary stasis. At the same time, it's kind of the best we have at the time being for how do we connect these paleo records to notions of modern day adaptation. So when we look at these papers that we recovered, we inferred that the coldest months are probably between 3.5 and 5.8 degrees centigrade for this particular location for this particular time. We also found that there are several frost-intolerant taxa that survived at that location, further indicating that it's probably not a freezing temperate habitat type. So this is the type of information we can collect from the fossil record to help inform what are the actual living circumstances for a fossil taxon in its environment. What types of temperatures can it survive? We then repeated that for the 5 different fossil taxa that we have. And this gives us a sense of where do the fossils live, not just in geographic space, but also in terms of their ecology. The next tricky part is that these fossil taxa simply, they're fossil pollens. They don't contain a lot of diagnostic information to place them precisely within a phylogenetic context. Yet we are confident that these fossil pollen represent a viburnum lineage. We just don't know where it belongs. So it makes it difficult to inject that information about the fossil pollen into ancestral state estimates. So the solution we adopted is an integrative evolutionary modeling framework. In essence, we're going to combine molecular information, which describes, and we can use this to basically describe how the genes will evolve along the branches of the tree, geographical information for both living and extinct species, and also biome information for living and extinct species. Notice that for the fossil pollen, we have no DNA. This is typical for lots of fossil taxa, as I mentioned earlier. So it's really critical that we somehow average over all of the placements of this fossil pollen so that we don't overweight where we think that fossil should appear in the tree. And the idea here is that we're going to integrate over all of the possible, we're going to not only integrate information, but we're going to probabilistically integrate over all phylogenies in the analysis. So here's what one integrated history would look like. On the left-hand side, it shows the biogeographic history with respect to regions over time. And these little nubs here correspond to the geographical regions of the species. We can also look at the habitat types for the species over time. And these nubs here correspond to the biome states and the fossil taxa included in the analysis. Now, like I said, we're going to be doing this in an integrative sense on the statistical side, meaning we will, in practice, it's a joint Bayesian phylogenetic model where we include multiple data sets with multiple models of character evolution for each set of evolutionary evidence. Looking at the maximum clade credibility tree, just one of these trees, that's just a summary tree that represents all of that diversity that we saw in the reconstructions. And also focusing on the ancestral state estimates for the region, we can see that these fossil taxa, they contain a little bit of information about when did lineages first enter, in this case, yellow, North America. What's more interesting to me for this talk is these fossil taxa also give us some information about what habitat types ancestral viburnum lineages probably occupied. Many of these lineages are green, indicating warm temperate biome affinities. And we see that we have a fairly strong support for warm temperate origin for the entire group. If we mask that paleobiome information from the fossil taxa, so we say that these fossil taxa, in fact, they have no information about the biome affinity. We reconstruct a much more ambiguous ancestral state ancestral state with about 1/4 supporting cold temperate, 1/4 of the probability supporting tropical, and 1/2 supporting warm temperate. We can look at this a slightly different way, where we look at the proportion of lineages in different habitat types over time. In this case, when we include the fossil taxa, we find that the vast majority of species in North America preferred a warm temperate habitat type. And only recently, after about 20 million years ago, do we see these cold temperate lineages rise to prominence. We can do this over all different regions in the left column here. And we see a similar pattern of cold temperate habitat preferences increasing over time. So this is when we include the fossil pollen with their biome states. When we exclude that fossil pollen biome state information, we see much higher support for cold temperate origins, even in regions like Southeast Asia, which probably never had many cold temperate habitat types back in the past. Now, in terms of the second strategy that we adopted for this problem, I also want to point out that we know a lot about global climate patterns and also the distribution of biomes over time. So viburnums are about 55 to 65 million years old. Note here at 55 million years old, we hit the Paleocene-Eocene thermal maximum. This is thought to be one of the warmest periods in recent history, certainly during the Cenozoic. At that time, there were some cool, but virtually no cold or boreal northern forests that we associate with regions like Russia and Canada today. Only after the Oligocene do you really see these cold forests begin to expand both in the northern and southern hemispheres. So it seems unlikely that viburnum could originate in this biome that was virtually non-existent 55 or 65 million years ago. So we thought we would design a model that somehow incorporated local information about habitat availability while reconstructing these biome affinities. To do this, we needed to associate regional structures of biomes with different geographical regions. So for example, this is a fairly good representation of the distribution of tropical biomes today. You can see it's mostly equatorial. Depending on how you define a tropical biome, if you go back to the mid to late Eocene, you'll see that tropical biomes are much more expansive, certainly at greater positive and negative latitudes. we took information, I'll get to this in a moment, but essentially we adopted a model, we designed a model rather, where we could incorporate the availability of these biomes with respect to different regions. So here we have 6 regions: Southeast Asia, East Asia, Europe, North America, Central America, South America. And for each of those regions, we could assign assign whether a particular biome had a strong, weak, or marginal presence. Those are recorded along the diagonal. And we could also record to what extent are regions connected through that biome. Do they share a strong, weak, or marginal connection? So if we are looking at present-day biomes, we see that Central and South American tropical biomes are fairly separated between these regions when compared to Southeast Asia and East Asia. When we go back to the mid to late Eocene, we see much greater connectivity among tropical biomes. So we did this not only for tropical biomes, but also for warm biomes and cold temperate biomes for 8 epochs, 6 regions, and 3 different biomes, using a wide variety of paleobotanical records. During this first 4 epochs, we see that the 6 regions are highly connected through tropical biomes with some amount of warm temperate biome prominence and virtually no cold temperate biomes. During the second time interval, we see that the tropical biomes become fragmented with respect to regions. Warm temperates begin to connect to, they change in their connectivity, but we certainly see a rise in these cold temperate forests that we know and love today. These 2 different intervals of time are separated basically at 34 million years ago, at the beginning of the Oligocene, when global temperatures begin to cool and the world becomes much more dry. We can then take these different paleobiome reconstructions, as we've encoded them, and put them into a model for how characters might evolve. So this animation basically shows how might lineages both change between biomes in these block diagonal matrices in red, but also move between regions on the off diagonals in blue. And so what we're seeing here is that the rates of change can differ over time. The exact way that these rates change will be estimated from the data set itself. We then ran our analysis under 3 separate conditions. The first condition is one assuming that we have a full set of possible paleobiome structure hypotheses. The second condition is one assuming where we only consider modern day biome structures when reconstructing the past. And the last one is assuming that all biomes always exist over all time intervals. When we use the paleobiome, which is the richest model, we recover a warm temperate ancestry in this middle red color. But for the modern biome and null biome models, we get a very strong ancestral affinity with this cold temperate habitat type, which probably didn't exist 70 million years ago. When we look at the distribution or the proportions of lineages over time, we find that for the paleobiome model, we prefer a warm East Asian origin for the group. For the other 2 analyses, we also get East Asian origins, but instead prefer these cold temperate habitat types. Overall, we find that strategy 1, when we use our full paleobiome data set, and keep in mind, this is based on the paleobotanical record, but using different fossil information from when we use specific viburnum fossil taxa, we recover a very similar history of a warm, temperate ancestry for the group. When we ignore the fossil taxa, either at the paleobiome level or for the individual viburnum fossil taxa level, we tend to get these much colder ancestral reconstructions for where viburnum species originated. So this might just be my disposition, but I guess when faced with did they originate in a tropical or freezing biome, it seems like it's the middle ground. Neither. It might be this intermediate habitat type which experiences some freezing, which could partly explain the scalariform perforation plates, while also not being so cold so as to completely eliminate the possibility of having some tropical affinity. Hard to say. I'm basically out of time, but I was just going to briefly mention 3 challenges that I think these integrative analyses face. Fundamentally, we're thinking about how do species associate with one another and what can we learn from sets of species. There are major challenges in fossil taxonomy and systematics that are not readily addressable, and they require immense amounts of paleontological expertise to resolve. These come down to issues of how do you assign names to different fossil taxa and how do you propagate those names over history. In our viburnum analysis, we found, these papers here identified a large set of viburnum fossil taxa that basically had been unnamed or removed from the group and no longer were viable representatives as viburnum. And this is not rare in paleontology, as far as I'm aware, where the fossil taxa, as you learn more about different morphological variants, they may end up being moved to entirely different groups over time. Another issue is that there are major biases in terms of where do we actually collect fossil taxa. Where are species actually fossilized? That depends on the underlying environment and geology. Where do we actually excavate these fossils? What countries tend to describe them and publish them? All of these factors really shape the way that paleontological information comes into scientific understanding. And it's not just at the level of natural processes, it's also at the level of sociological processes. How do we publish? Who do we cite? Things like that. The last slide is, of course, there's some major issues in terms of associating how we think about species concepts and speciation when working between what you might consider neontological and paleontological scales. Of course, at the neontological scale, we have the benefit of being able to potentially try to cross species, see if they can actually reproduce. We can use genetic information to understand how alleles are exchanged between lineages and so forth. Look at behavior, look at chromosome structure, all of that. With fossil taxa, we're normally restricted to simply geographical information, morphological information, and perhaps extrapolations based on what we know about developmental biology. I would also say that, when I think about neontological perspectives on species and speciation, there are some interesting ideas that crop up in paleontology that I don't see appear in microevolutionary studies that much, such as what is the definition of a species as an entity that persists over time? It's a bit of a ship of Theseus problem. After what extent of time, if a species is changing, does it remain that species? At some point, is it no longer the same species with the same identity? Does it deserve a new name? Paleontologists would call that an anagenetic speciation event that begins with the pseudo-extinction of the previous lineage. I think also in microevolution, depending on the framework you're using, we also have different ideas about how does speciation actually occur. There is a lot of controversy, and I think there's still some amount of unresolved controversy in terms of to what extent do models of punctuated equilibrium are they both explained by microevolutionary models and completely satisfactory to accurately predict the way that paleontological patterns are produced. There's a difference there between is a microevolution sufficient to explain all macroevolutionary patterns versus can we do that with high accuracy? So I guess that's a little controversial tidbit I'll leave off for the discussion later. I don't know where I stand on it personally. With that said, seeing as how I've gone over time, I'd just like to thank you for your patience. I thank the steering committee for inviting me to speak, and thanks for Josef for sharing his wonderful thoughts on the topic.
Thank you so much, Michael. So in the interest of time, we're going to open it up to more general discussion. And I wanted to start with